As for studying diseases and disease mechanisms, it's important that we understand genetics. When we think about genetics, we think of DNA and genes, but actually we also need to consider the proteins for which the genes encode. It is these proteins that form cell structures and control how the genes function. So I want you to read Chapter 5, and we're going to review some of the genetic material that you should remember from A&P. DNA is the basic substance of which chromosomes are made. DNA has three basic components: the sugar molecule, deoxyribose, a phosphate molecule, and four types of nitrogenous bases. Two of these bases, cytosine and thymine, are single carbon nitrogen rings, and the other two are double carbon nitrogen rings. The other two are called adenine and guanine. Because of their shape, this means they only combine in certain ways. Adenine only combines with thymine, and cytosine only combines with guanine. The order of combining can be reversed. We can have adenine first and thymine second, or we can have thymine first and adenine second. We call these bases that combine together complementary bases. So adenine and thymine are complementary bases. Cytosine and guanine are complementary bases. Bases compose the crossbars of the DNA molecule. The sides of the double helix are made out of phosphates and sugars. The sugars is deoxyribose sugars. Bases are found in the nucleoplasm as part of a nucleotide. A nucleotide is a basic unit that consists of a base, a phosphate, and a sugar. And it's these nucleotides that are linked together to make a DNA molecule. The bonds between the bases are hydrogen bonds. And hopefully you remember that hydrogen bonds are fairly weak bonds that can be broken easily. We have the same type of bonds in proteins. When they are broken, the protein is denatured, which means it loses its original characteristics. One of the main things that DNA does is it directs the synthesis of the body's proteins. These proteins are made out of amino acids, and there are 20 different amino acids that are found in the body. Yet, there are only 4 different bases in the DNA molecule. So this means we cannot have one base being the code for one particular amino acid. Using a 2-base pair sequence would give us 16 possible combinations, but that still wouldn't be enough to code for 20 different amino acids. Using a 3-base pair sequence gives us 64 possible combinations, and this is how amino acids are coded. A 3-base sequence on the DNA molecule represents one particular amino acid. These 3-base sequences are called triplets or codons. There's a chart in your book that shows the various codes that have been worked out for the different amino acids in our body. 3 of the codons are termination codons, and the other 61 codons code for specific amino acids. Most amino acids are specified by more than one codon, and again you can refer to that chart. In order to make a protein, amino acids must be strung together in a particular order. This order is directed by the DNA. If there is an error in the DNA, such as a mutation, there will be an error in the protein. If we do not form a needed protein, we will be unable to carry out some process or form some cell component. Or, if we lack a particular amino acid due to poor nutrition, we will also be unable to make a needed protein. DNA is a universal genetic substance. All living organisms use the same DNA codes to specify proteins. The only exception to this is that mitochondria have their own DNA patterns. Some of the mitochondrial codons encode for different amino acids than the same nuclear DNA codons. We're not going to worry about mitochondrial DNA in this course, but I just simply wanted you to know that it does exist. Besides having the ability to specify amino acid sequences, DNA must be able to replicate itself accurately during cell division if it is to serve as the basic genetic material. DNA replication involves the breaking of the weak hydrogen bonds between the bases, dividing the DNA into 2 strands of single bases. The unpaired bases then attract their complementary base, and so we end up with 2 identical strands of DNA. Several proteins are involved in this process. One unwinds the double helix and another holds the strands apart. Another protein, DNA polymerase, is an enzyme that connects the correct nucleotides to the free end of the new strands. DNA polymerase does some proofreading as well as connecting. If the wrong nucleotide has been inserted, it is removed and a correct one put in its place. This really enhances the accuracy of DNA replication. In addition to DNA, there is another very important genetic chemical, and that is called RNA, or ribonucleic acid. RNA is a type of nucleic acid that is very similar to DNA. It has some of the same bases. It has ribose sugar instead of deoxyribose sugar. It has uracil as a base instead of the base thymine. RNA is a single helix. In the picture, you see DNA on the left and RNA on the right. While DNA replication takes place in the nucleus, protein synthesis is going to take place in the cell cytoplasm. The DNA molecule is too large to pass through the nuclear pores and go to the ribosomes, so something else must carry the code or pattern from the nucleus to the ribosome. The transport of the genetic code to the cytoplasm is done by RNA, which is made through the process of transcription. Transcription involves the making of a strand of messenger RNA. Ribosomal RNA makes up part of the ribosome, and transfer RNA will be discussed soon. The process of synthesizing RNA from a DNA template is called transcription. The result of transcription is the formation of messenger RNA. RNA polymerase is an enzyme that bonds to a site on the DNA, which is the beginning of a gene. It pulls the DNA strands apart from one another. Now, it's not opening up the entire strand, but it's just simply opening up one area along the DNA strand. One of these separated strands will serve as a template for the sequence of messenger RNA nucleotides. The bases of messenger RNA are complementary to those in the DNA template strand. Uracil is substituted for thymine, and except for this, the messenger RNA is identical to the strand of DNA which is not serving as the template, the strand it is temporarily replacing. Transcription continues until a termination sequence is reached. Then the RNA polymerase detaches, the messenger RNA is released, and the two DNA strands reconnect. The messenger RNA, because it's a single helix, is small enough that it can pass through the pores in the nuclear membrane and enter the cytoplasm where it attaches to a ribosome. Once there, the next step, which is called translation, can take place. Translation is the process by which RNA directs the synthesis of a protein. Translation takes place at the ribosome, and what happens is the ribosome passes along the strand of messenger RNA, reading it and reading the sequence of amino acids that need to be taken into place. This process also involves transfer RNA. Transfer RNA consists of a clover-shaped strand of about 80 nucleotides. One side has a place where there's an amino acid can attach. The other side has a sequence of 3 nucleotides called the anticodon. The bases in the anticodon will pair with the complementary bases in the messenger RNA. Each type of transfer RNA is specific for a single amino acid. Ribosomes are made up of equal parts of protein and ribosomal RNA. Ribosomes are formed in the nucleus, and then they move out into the cytoplasm, where they're found either floating free in the cytoplasm or attached to the rough endoplasmic reticulum. The ribosome binds to a piece of messenger RNA, and the ribosome then moves along the messenger RNA sequence, codon by codon. As each codon is processed, you remember a codon is a group of 3 bases. As each codon is processed, an amino acid is brought into place for the transfer RNA. An enzyme causes peptide bonds to form between the amino acids. This continues until the termination signal is reached. This is simply like stringing together a necklace made out of beads. And if you want the beads in a certain order, the messenger RNA is telling you the order that the beads need to be put in to. So if you were going to make a necklace that had your name on it, the amino acids would be strung together so that your name would be revealed. In a protein, the sequence of amino acids is of vital importance. Just as if you were making a bead bracelet or necklace showing your name, the beads must be in the correct order. So it is with a protein. If you don't have the amino acids in the correct order, you don't have the right protein. You must have the correct amino acid in the correct sequence. Be sure you know the difference between transcription, which is the formation of the messenger RNA, and translation, which is the formation of the protein molecule at the ribosome. Sometimes there's an error in our genetic material. We call these errors mutations. A mutation is an inherited alteration of genetic material, or it's a change in genetic material that can be passed on to your offspring. Mutations are classified according to where they occur. Do they occur in the body cells, or do they occur in the reproductive cells? If mutations occur in the body cells, the cells other than the gametes, we refer to them as somatic mutations. Cancer is an example of a somatic mutation. If your parents develop skin cancer before you're conceived, that skin cancer isn't passed on to you. If the mutation occurs in the gametes, then we call it a germ mutation, and these are the types of mutations that may be inherited. Some mutations are very drastic, others are barely noticeable. There are several different things that we know will cause mutations. One of those is radiation. Radiation forms electrically charged ions that produce chemical reactions that can change the DNA basis. So whether you're exposed to the radiation from the sun, the ultraviolet light, or whether you're exposed to what we might call nuclear radiation, this can change the sequence of your DNA. There are several different chemicals that will change the bases making up the DNA or interfere with the process of base pairing. Some examples of these chemicals are thalidomide, alcohol, Accutane, some of the cancer drugs, formaldehyde, sodium nitrate, and vinyl chloride. There are several more, but these are all that I'm going to list at the time. Vinyl chloride is used in making PVC pipe and in some packaging. Then we also know that sometimes viruses will cause mutations. The measles virus, herpes virus, chickenpox virus, all have been known to cause mutations. Other mutations are linked to maternal disease or altered metabolism of the mother. Maternal hypertension or folic acid deficiency during embryonic development has been known to result in mutations with the offspring. And sometimes we simply have spontaneous mutations, mutations that occur for no known reason. Let's think a little bit about chromosomes. The human body contains two types of cells, gametes, which would include egg and sperm cells, and somatic cells, also known as body cells. Somatic cells are diploid. This means they have 46 chromosomes, or sometimes we think of this as 23 pairs of chromosomes. One member of each pair comes from each parent. These cells are formed by mitosis, meaning somatic cells. Gametes, on the other hand, are formed by meiosis, and gametes are haploid. They have only 23 chromosomes total. They have one member of each of the 23 chromosome pairs that are found in humans. Homologous chromosomes are the two members of each pair. Homologous chromosomes are typically nearly identical in appearance, but they contain genetic material for the same traits. The members of a homologous pair are called chromatids, and you get one chromatid or chromosome of a homologous pair from each parent. So, for example, in chromosome pair number 5, one of those chromosomes came from your mother, the other one came from your father. Centromeres are an area on the chromosome where, when chromosomes are undergoing replication, the chromosomes are attached to each other. The location of the centromere determines the length of the arms in each chromosome. So during mapping, regions on the arms are identified. Human cells normally have one pair of sex chromosomes and 22 pairs of non-sex or autosomes. Mitosis is the normal type of cell division that produces two identical cells. However, gametes are formed by a process called meiosis. During meiosis, there is one replication of the chromosomes, and then there are two divisions of the chromosomes in the cytoplasm. This process reduces the chromosome numbers in gametes. So, while somatic cells have 46 chromosomes, gametes, after they've been produced by meiosis, will only have 23 chromosomes. If a mistake occurs during this process, we can end up with an extra chromosome in a gamete or being short a chromosome in a gamete. Sometimes we also can end up with an entire extra set of chromosomes in a gamete. And of course, any deviation is going to cause problems if that gamete happens to be involved with fertilization. Maps of chromosomes are referred to as karyotypes. On this slide, we see a karyotype of a male. If you look at it closely, you can see there's one X chromosome and one Y chromosome. On the end of each chromosome, there's a small area of DNA called the telomere. And it's theorized that each time a cell divides, a small amount of DNA is lost. And so we think that over time, this loss will inhibit cell replication. That over time, cells lose their ability to replicate and replace damaged and aging cells. And this is one of the reasons why we don't live forever, because we lose some of the DNA from our telomeres every time our cells divide. Let's review some genetic terminology. The position of a gene along a chromosome is known as its locus. Different forms of the same gene are called alleles. So, for example, we have an allele for, we have more than one allele for eye color, so we might have a brown allele and a blue allele. Phenotype is the appearance of the gene. Is the person tall or short, blonde or brunette, blue eyes or brown eyes? Genotype refers to the actual genetic makeup of the gene. And when we talk about genotype, then we talk about homozygous and heterozygous. If both genes for a characteristic are the same, we use the term homozygous to describe the genotype. So you could be homozygous for being able to roll your tongue. If the two genes for the characteristic are different, then we say the individual is heterozygous, meaning that it has one of each kind. Now, don't get homozygous confused with homologous. Homologous refers to chromosomes, which are members of the same pair. Homozygous means that both of the genes you possess for a characteristic are the same. A carrier is a person who is heterozygous for a recessive trait. They do not express the recessive trait, but it can be passed on to their offspring. Genome mapping will revolutionize medicine, we hope. The Human Genome Project was started in 1990 by, among others, James Watson. The basic work on the project was completed in 2003, but there's still a lot of research and analysis going on, and there are still some regions of the genome that are unknown. The current estimate of 25,000 to 30,000 genes in the human genome surprised scientists who had been predicting for many years that there would be over 100,000 genes present. Of these genes in the humans, another thing that was surprising was to discover that 99.9% of the human genes are held in common with the rest of the human race, meaning that only 0.1% is what makes a person unique. Many of the genes causing genetic disorders have been discovered or are being investigated. Recombinant DNA techniques are being used to produce proteins such as insulin, which are identical to the insulin produced by human cells. Gene therapy techniques are being developed that use a virus to deliver genes to the cells of humans and other animals, as well as plants. This therapy has been used with limited success, and there are still many problems to solve before we arrive at the place where we can insert normal genes in all newborn infants who are born with defective DNA. Sometimes what we need is not to replace defective genes, but to simply keep defective genes from operating. Research is being done on how to use pieces of RNA to suppress the expression of abnormal genes that a person might possess. We already know that genes are composed of sequences of DNA. An error in one gene may lead to a recognizable genetic disease. There are more than 15,000 genetic conditions that have been identified. As infectious diseases have come under control, 1/3 of the beds in pediatric hospitals are occupied by children with genetic diseases. In addition, many common diseases that affect adults, such as hypertension, coronary heart disease, diabetes, and cancer, are now known to have important genetic factors. Great progress is being made in the diagnosis of genetic diseases and the understanding of genetic mechanisms. Genetics is one of the most rapidly advancing fields of medicine. Congenital defects are abnormalities which are present at birth, as opposed to acquired defects which are caused by events after death. Congenital defects may be caused by genetic factors as well as environmental factors. Genetic defects can be caused by DNA errors or by changes in the arrangement of the DNA on the chromosome. First of all, we're going to talk about some chromosome disorders. Chromosome disorders account for a large portion of mental retardation and spontaneous abortions that occur. What we see here is a karyotype of an individual with normal chromosomes. Such a karyotype consists of 23 pairs or 46 individual chromosomes. Pairs 1 through 22 are autosomes. Autosomes carry more genetic information than the sex chromosomes. Pair 23 are the sex chromosomes. When we are discussing the chromosome number of a person, we normally would write this 46XX if they're female, or 46XY if they're male. Chromosomal disorders are usually due to defects in meiosis. When we're talking about chromosome numbers, there's some terminology we use. Cells that have the normal number of chromosomes are said to be euploid. The prefix eu means true or good in Greek. If cells have more than the diploid number, they are said to be polyploid cells. Normally, a few tissues, such as the liver and bronchial and epithelial tissues, may have polyploid cells, but that is not extremely common. If a zygote has 3 copies of each chromosome rather than the usual 2, it is said to be triploid. The chromosome number would then be 69 rather than 46. If a zygote has 4 copies of each chromosome, it is said to be tetraploid, and the chromosome number would be 92. Almost all triploid fetuses spontaneously abort or are stillborn. A few of them have survived to term, but they die soon. Tetraploidy has been found mostly in early spontaneous abortions, but a few humans have been born with the tetraploid condition. However, they do not survive. These two conditions, tetraploidy and triploidy, probably account for about 10% of all known miscarriages in humans. An aneuploid cell is one that does not contain a multiple of 23 chromosomes. If the cell has an extra chromosome, we call this condition trisomy. There will be 3 copies of one of the chromosomes. Some newborns with trisomy will survive. Those that have trisomy 8 or 13 or 18 or 21 are the only trisomies that show up with any frequency. Trisomy 16 is often found among miscarried fetuses, but is not seen in live births. Monosomy is when an individual is short 1 chromosome. If the missing chromosome is an autosome, this never results in a live birth because there simply has been too much genetic material lost. If the missing chromosome is a sex chromosome, the child probably will survive unless it is just a Y with no X. However, there may be some abnormalities. Details will follow about that. In general, loss of chromosome material has more serious consequences than duplication of chromosome material. The best known example of trisomy is Down syndrome. Down syndrome, of course, is trisomy of chromosome number 21. Down syndrome was first described in the 1860s. It is believed to be the most common form of a chromosomal disorder. Down syndrome is seen in approximately 1 in 800 live births. A person who suffers from this trisomy will be mentally retarded. Their IQ is usually somewhere from 25 to 70. They will be short in stature. They will have a protruding tongue, flat, low-set ears. Average life expectancy for an individual with Down syndrome is 60 years. Approximately 10% of these children die before the age of 10. In the United States, about 10,000 infants with Down syndrome are born each year. Research has shown that oocytes will change during the aging process, perhaps due to environmental exposure. And so there is an increased occurrence of Down syndrome as the mother ages. For people who are 25, there's 1 chance in 1,300 of having a child with Down syndrome. For people who are 45, that goes down to 1 chance in 30. Most of the cases of Down syndrome are the result of non-disjunction. People who have Down syndrome have an increased risk for leukemia, Alzheimer's, and heart disease. Here's the karyotype of a male child born with Down syndrome. Notice the extra 21st chromosome. This is also a good place for you to notice the difference between the Y chromosome and the X chromosome. Now this is the normal difference that we would see in any normal male individual, but I just want to point out that the X chromosome contains quite a bit more genetic material than the Y chromosome. I just told you that Down syndrome was the result of non-disjunction. Well, non-disjunction is an error that occurs during mitosis or meiosis. It's an error when homologous chromosomes fail to separate. And in meiosis, this will produce some gametes that will have 2 copies of a chromosome and others that will have no copies of the chromosome. So if non-disjunction occurs during the formation of gametes, we'll get some gametes that have 24 chromosomes and some gametes will only have 22 chromosomes. If one of these gametes unites with a normal gamete, the result is a zygote that is either trisomic, which they have 1 extra, or monosomic, which they are short that 1 chromosome. Here are some examples of non-disjunction showing the results of chromosome number abnormalities. On the left, non-disjunction occurred during the first meiotic division. On the right, non-disjunction occurred during the second meiotic division. Either way, the offspring were abnormal. Another cause of abnormality in chromosomes is translocation. Translocation is the interchanging of genetic material between non-homologous chromosomes. A reciprocal translocation occurs when the break takes place between 2 different chromosomes and the material is exchanged. When this happens, the person is usually normal since there is still a normal amount of genetic material. However, the gametes may or may not show duplications of genetic material or deletions of genetic material. Translocation will introduce genetic variety. It may cause abnormality in children, or it may not. Depends on how much material is exchanged. If this break happens near the centromere and the long arms fuse together, we can get Robertsonian translocation, which we're going to see in the next slide. The most important type of translocation from a clinical standpoint is the Robertsonian translocation. In this translocation, the long arms of 2 non-homologous chromosomes fuse at the centromere, forming a single chromosome. Robertsonian translocation is found only in chromosomes 13, 14, 15, 21, and 22 because the short arms of these chromosomes are very small and they contain no essential genetic material. When a Robertsonian translocation takes place, the short arms are usually lost during subsequent cell divisions. Since the carriers lose no important genetic material, they are normal, although they only have 45 chromosomes in each cell. So they do fine. The problems arise for their offspring. For example, if there's been a fusion in the short arms of chromosomes 21 and 14, an offspring inherits a gamete containing the fused chromosome, receives an extra copy of the long arm of chromosome 21. This person then would develop Down syndrome. Robertsonian translocation is responsible for about 3 to 5% of the cases of Down syndrome. Another type of change which involves the exchange of genetic material is crossing over. Crossing over usually does not present problems. Usually it simply gives us more genetic variety. Crossing over usually happens during metaphase and anaphase, when the chromosomes are in the middle of the cell and they will twist around each other, breaking and then refusing. However, when this occurs, it is possible to lose a portion of the chromosome if it does not refuse, and so that could give us some difficulty. If material is lost, then we refer to that as the deletion. Deletions happen when chromosomes break, as in crossing over, and some DNA is lost. Even if the chromosome unites with a normal gamete to form a zygote, often so much material is lost that there may be serious consequences. The most often cited example of a deletion is the Cry-de-Chat syndrome, in which there are deletions in the short arm of chromosome 5. The name Cry-de-Chat means cry of the cat, and this describes the characteristic cry of the affected child. Symptoms of a child with this syndrome include low birth weight, severe mental retardation, problems feeding, severe speech and motor delays, and severe behavior problems such as hyperactivity. 1 in 50,000 births may involve Cry-de-Chat syndrome. 80% of the time, the deletion is in the male-provided gamete. Now I want to talk a little bit about sex chromosome disorders. The incidence of sex chromosome aneuploidies is fairly high. Among live births, it occurs 1 in 400 times for males, 1 in 650 times for females. Because sex chromosomes contain less genetic material than autosomes, these conditions are usually less severe than autosomal aneuploidies. All forms of sex chromosome aneuploidy, except for Y with a complete absence of X chromosome, will allow for at least some of the individuals to survive. Individuals who simply have aneuploidy with only a Y do not survive to be born because there's too much loss of genetic material. One of the most common sex chromosome aneuploidies is trisomy X. It affects about 1 in 1,000 newborn females. Often these people are undiagnosed because there's usually no noticeable physical abnormality. However, sterility, menstrual irregularity, and some mental retardation may sometimes occur. Klinefelter syndrome is another example of a sex chromosome aneuploidy. In this syndrome, the individual will have 2 X chromosomes and 1 Y chromosome. Because of the X chromosome, they will be male in appearance. However, they usually are sterile. Many times these people will not be recognized as having any abnormality. Sometimes they have learning disabilities. Sometimes secondary sex characteristics are slow to develop. That is often treated with testosterone. Another example of sex chromosome aneuploidy is Turner's syndrome. In Turner's syndrome, it's monosomy X. The individual is always female because they simply have 1 X chromosome and no Y chromosome. They are not mentally retarded, but they may have some impairment of their mathematical reasoning ability. They tend to be short in stature. They will have some Health concerns, many times they will have congenital heart disease. Often they have an underactive thyroid. They may have a tendency to develop diabetes and autoimmune disorders. They typically are treated with estrogen to promote the development of secondary sex characteristics, and also with growth hormone because of their tendency to have short stature. They are infertile, but successful pregnancies have occurred in people with Turner syndrome using in vitro fertilization techniques. It is estimated that only about 1% of the people who are conceived with Turner syndrome actually survive to full-term birth. This diagram is simply showing the possibilities of having Turner's or Klinefelter syndrome. On the left side of the chart, we see normal meiosis occurring. On the right side, we see non-disjunction that occurs in the ovum. When the ovum that is the result of non-disjunction joins with a normal sperm cell, you can see the 4 possible outcomes. The last form of sex chromosome aneuploidy is the condition XYY. These people, of course, will be male, and this condition occurs in about 1 in 1,000 live births. XYY is the result of non-disjunction in the sperm cell. And the reason we know that it was non-disjunction in the sperm cell is because we ended up with 2 Y chromosomes. If it had been non-disjunction in the egg cell, we would have ended up with 2 X chromosomes. There's a lot of variation among individuals that have this condition. Many of them have no significant issues and often are unaware of any genetic abnormality. A few of them may have delayed motor skills, language issues, learning disabilities, or sometimes emotional difficulties. In the past, they thought that having the extra Y chromosome could cause these individuals to be aggressive or display violent behavior. This theory has been absolutely disproven.